Hi guys, I am Joyce Magustin. Together with the group 3, we are gonna tackle the manipulation of compound of microscope. So first, I am gonna tackle about the functions and parts of the microscope. So first, we have the eyepiece. The eyepiece lens magnifies the image, while the body tube holds the eyepiece. Arm holds the microscope. Stage height adjustment is to allow the long high-power lens to fit over the slide. The coarse adjustment is for rough focusing, and fine adjustment is for precise focusing. While clips holds the slide. Nosepiece revolves to move to desired lens into position. Diaphragm lever controls the amount of light reaching the objects. Objective lens, it magnifies the image. Condenser, it focuses light on the object may not be present in all microscopes. Mirror or light source, it supplies light to the object. And lastly, the base, it holds the entire assembly.
Hello everyone, I'm Cherry Kate and I'm going to discuss how to calculate the total magnification. So our learning outcome is how to calculate the total magnification. But first, what is the difference between magnification and the total magnification? When you say magnification, it is the process of enlarging the size of an objective as an optical image. While total magnification, it is a compound microscope. The total magnification is the product of an objective and ocular lenses. The magnification of our ocular lenses on your scope is equivalent to 10x. So this is the formula. Magnification equals to our eyepiece power. When we say eyepiece power, it is the ocular lenses. So ocular lenses is given na it is equivalent to 10x. So the eyepiece power will be multiplied to objective power. So what is objective power? Objective power, it is the size of the image. So as easy as that. And eyepiece power multiplied by the objective power. So for example, and we have the low power. And the low power, it has the eyepiece power 10x multiplied by its objective power 10x also. So 10x multiplied by 10x is equal to 100x. And also we have a high drive. And for example, and the high drive has its eyepiece power 10x as usual and multiplied by 40x also and 10x multiplied by 40x and it equals to 400x. As easy as that, just multiply the eyepiece power and the objective power, so you will be having that total magnification. That's all. Thank you.
My report is all about guidelines in using the microscope. Everything on microscope is unbelievably expensive, so be careful. Number 1. Hold a microscope firmly by the stand only, never grab it by the eyepiece holder. Number 2. Hold the plug, not the cable, when unplugging the illuminator. Number 3. The bulbs are expensive and have limited life, only turn off when not in use. Number 4. Always make sure the stage and lenses are clean, putting the microscope away. Number 5. When cleaning the microscope, use lens cleaner. Number 6. Cover the instrument with a dust jacket when not in use. Number 7. Focus smoothly. Don't try to speed through the focusing process or force anything. Number 8. If not working, don't try to fix it. Ask someone who knows it.
Morning everyone. My report is all about the guidelines in recording observation. If we are inside the lab, we can see the specimen using the microscope. We can label it and give the magnification. But since we are not inside the lab, it is okay that there is no total magnification. The important is you know how to compute it. The question is, how we compute the total magnification? In a compound microscope, the magnification is going to be determined by the magnification of objective as well as the magnification of eyepiece, which is 10 times. To know the magnification of objective, you will simply mean to observe the magnification of the power that is indicated on it. For example, 4x, 10x, 20x, and 40x objectives. In order to calculate the total magnification, you need to multiply the magnification of objectives to magnification of eyepiece, which is 10 times. For example, you are using the 20 times of your objective. Your specimen will appear 200 times larger than with the naked eye. That's how we record the observation.
Good day everyone, my name is Christine Dana Belmones and I am discussing about amoeba. Amoeba is a protozoa. It has irregular shape, unicellular organism. Amoeba also has digestive vacuole, food vacuole, nucleus, contractile vacuole, pseudopodium, ectoplasm, endoplasm, and we have also the cell membrane. Amoeba helps to remove the waste from the body, and amoeba can be transmitted by food and drinks. That's all about amoeba, and thank you.
Paramecium. Paramecium is a single-celled protozoan eukaryotic cell, and it basically found or swim in streams in the environment. As we can see in the illustration, Paramecium is surrounded by cilia, and cilia has two main purposes. First is motility, locomotion or moving around, and the second one is food capture. And remember that the cell wall of Paramecium helps to protect the cell from swelling from picking up too much water. And the Paramecium has two contractile vacuole, and it was surrounded by array, basically act to collect water. And it also has two nucleus. The first one, the larger one, is macronucleus. It is responsible for metabolic and none other reproductive function. And the second one, the smaller, is micronucleus. It is responsible for the cell, the actual cell division when it's time for the Paramecium to divide.
Hello guys, I'm Desiree Beloria and I'm discussing about Euglena. Euglena, unicellular organism that belongs to the genus Protis, such as they are not plants, animals, or fungal. In particular, they share some characteristics, both plants and animals, or can manufacture their own food characteristic that's seen in plants. They're also capable movement and consuming, which characteristic of animals. Do they unique characteristic? There are some debates as the phylum should place. Historically, they are being classified by scientists. Euglena lives in the fresh and blackish water, rich in organic matters, and can also found in the moist soil. Euglena are characterized by an elongated cell with one nucleus, numerous chlorophyll containing chloroplast and contractile vacuole, an eye spot, and one or two fragla. Unlike plant cells, Euglena lack of liquid cellulose wall and have a flexible pellicles that allows them to change shape. Though they are photosynthetic, most species can feed heterotropically and absorb food directly through the cell surface via phagocytosis. Food is often stored as specialized complex carbohydrates known as paramylon, which enable to organisms to survive in low light conditions. Most Euglena have a life cycle consisting of pre-swimming stage, euglena produced rapidly by the types of asexual production method known as binary fission. The euglenal cell produces its organelles by mitosis and then split longitudinally into daughter cells. And when the environmental condition become unfavorable and too difficult for euglena to survive, they can enclose themselves within a thick wall productive cyst. Productive cyst formation is characteristic of the non-bottle stage. And that's all for the euglena. Thank you.